So how does the stomach's acidic environment aid in digestion? Well, the acid in the stomach ranges anywhere between a 1 to a 3 on the pH scales. That's going to be a strong acid. What that acid is going to do is start to break down macromolecules into smaller particles, so the body will be able to chemically and mechanically digest that, and then it can take those particles and then use them in the body after it's absorbed to rebuild into things that the body needs. So along with muscles that are squeezing and squishing and moving things around, this acid is actually going to help break down those things chemically as well as mechanically. Does reducing the stomach's acidity have health consequences? Well, they have found that when evaluating this, acidity-reducing medications increase the potential for food allergies. So this is one thing that they want to study and look at. They're going to do 2 experiment strategies. With these, the first, they're going to evaluate the effectiveness of protein digestion enzymes at several different levels of acidity. Now remember, with that acid inside of the stomach, its job is to break down that food so then you could use those macromolecules in smaller forms to rebuild into things that you need. But with that, if you have too much acid inside of your stomach, then and not enough food, or you don't have a protective lining, it can eat through it and actually cause ulcers or cause other problems. So they're worried about reducing the acid because you need that to break down the food, but we also don't want to deal with ulcers. So results, we have a typical stomach acid of pH 2. The proteins in milk are going to be completely digested within 1 minute. So that's going to be the normal control. That's the regular acid in the stomach. At least the acidic pH levels of 3 and 5, they're going to go in and look, and that milk protein is going to be largely indigested after an hour. So once you drink milk, once it goes into the stomach at a pH of 2, it will break it down within 1 minute. At a pH of 3 to a 5, it's going to be indigested even after an hour. So from these results, what could the researchers conclude about the role of stomach acidity in digesting proteins? Well, the acid does need to be stronger in order to break these down. In their second experimental strategy, the researchers observed 152 people who were seeking treatment for an ulcer before and after a 3-month antacid treatment. They evaluated each patient for signs of allergic reactions to a variety of proteins. The results? Before the 3-month antacid treatment, 10% of these subjects will exhibit signs of having an immune response to at least one of 19 common dietary proteins that they're going to test for. But after the 3 months of antacid treatment, they find that 26% of these subjects are now going to start exhibiting signs of having had an immune response of at least one of these 19 tested dietary proteins, if not more. So from these results, what could the research team conclude about the role of antacid medications in causing normal dietary proteins to stimulate an allergic reaction? Well, we need that higher or lower acid rate, the stronger acid, in order to break these proteins down. When they're not broken down and they move through the system, the body's immune system is going to recognize those particles as being something that is not belonging to the body. So it's going to want to get rid of it, and it's going to create an autoimmune response, and it's going to attack those. This is actually going to create an allergy. Now, as you get older, your body does start to reduce the production of lactase enzymes, and so a lot of adults will become lactose intolerant. But also, if you've raised the pH of your stomach by taking antacids, it could also be that this product simply isn't being broken down properly and your body's reacting to it with an autoimmune response. So summary, dietary proteins are not digested by the stomach when the stomach's pH is increased as a result of medications taken to treat ulcers, heartburn, and other digestive problems. This, unfortunately, can put people who take an antacid medication at risk for developing an allergic response to common foods. Going into our macromolecules section, the first macromolecule we are going to cover is going to be the carbohydrates. The carbohydrates are what we take in most of our diet. So this will be the larger portion of foods that we eat, anywhere from 30 to 40%. Carbohydrates are going to be made up of a monosaccharide as its subunit or base unit. So that's going to be the smallest particle that you're going to find this in. If you've ever had a low-carb diet, then you realize that cutting carbs from your diet will cut a lot of your energy molecules. So you're going to have to get energy from other things. So what things would you think that you would get energy from? Well, with these types of diets, you're going to get most of your energy from proteins and fats. If you have a high-carb diet, you're going to cut these proteins and fats and get most of your energy from carbs. We're going to discuss carb loading, where you take in lots of carbs before an event. And we're going to discuss fiber intake too. So all of these are going to be huge health topics that you'll hear about in the news and in lots of literature. But why are these carbs so important or not important in the diet, depending on which one that you're talking about? So like we said, carbs are going to be a macromolecule that the body takes in, and is going to be made up of smaller blocks called monosaccharides. With other macromolecules we're going to cover lipids, we'll cover proteins, and we'll cover nucleic acids. These are all macromolecules as well, and we'll go over their smaller particle or unit that they're broken into. So like we said, carbohydrates, lipids, proteins, and nucleic acids are what we're going to cover. So with the carbohydrates, they're made up of mainly carbon, hydrogen, and oxygen. So the CHO part of the CHON that we talked about. They're going to have the same number of carbons as waters. They're also going to be the primary fuel for organisms. They are found in our cell structures. They help as recognition particles that are attached to proteins, and we call those or refer to those as glycoproteins that are embedded within the cell membrane. They are, like I said, the subunit of monosaccharide, which is going to be a one sugar molecule. That's what it's going to be made up of, and you'll hook those together with these energy bonds in between, and then you can make disaccharides, or you can even make polysaccharides, which is going to be many of these sugars hooked together. So looking up there in the picture, you'll see a lot of foods that we refer to as our carbohydrate foods. With these foods, that energy is going to be found in the bond. So when you take in these carbohydrates as fuels, your body is going to start breaking them down within the mouth when you start chewing them up. Your salivary glands are going to release amylase and other enzymes to help break these up. Once you swallow it, it goes into the stomach. The stomach's then going to proceed to continue to break it up within that stomach acid and by movement of the smooth muscle. The carbon and hydrogen bonds are then going to be easily broken, and the organism can capture that energy and release it when the bond's broken and put into use. So it takes that energy and it restores it in other chemical bonds within the body, mainly in particles called ATP. So carbohydrates are going to include macromolecules that function as food. Glucose provides energy for the body's cells. So glucose is going to be a simple sugar. It is a monosaccharide, and it's what we break those sugars that are in the carbohydrates down into. So the simple form of sugar. Once your body uses what it needs, it can then store up to about 3 days worth of glucose in the body, and it stores it in the liver and in the muscles in the form of glycogen. Many complex carbohydrates are going to be time-release packets of energy. So when you take in a complex carbohydrate, it's going to be many of those monosaccharides hooked together, so we'll refer to it as a polysaccharide. And then it's going to break those down over a period of time, so it takes a while to break it down. And so that's why it's considered to be complex, and that's why you would want to eat a complex carbohydrate before a big event. And that would kind of be your carb loading, taking all those in. But not all carbohydrates are going to be digestible. Some are going to just act simply to help push things through the system, such as fiber. So with those monosaccharides, we said that was the simplest form of sugar. It will contain 3 to 7 carbon atoms. Plants make this in a form of glucose. Once they have used what portion of glucose they need, they can store this for later, the same as people. They store it as starch. So plant storage of glucose will be starch. The usable form is glucose. Fruits and vegetables will actually create sugars that are going to be a monosaccharide or a simple sugar form as fructose. So the suffix OSE tells us that it is a sugar, and of course all sugars are carbohydrates. Here's some other forms of monosaccharides. Know your 3 examples of monosaccharides. You're going to have glucose, fructose, and galactose. With these, notice that the chemical formula is all exactly the same. So it'll all be C6H12O6. It's simply how these molecules are put together that gives them their different characteristics. Most carbohydrates ultimately are going to be broken down into its smallest form, which is glucose. The glucose then circulates in your body at a concentration of about 0.1%. So it goes into the body. You're either going to use it as energy or it's going to be stored. If energy is needed, those molecules get broken down and are used by the cells, which get their energy. If it's not needed, it's then going to be stored. If it's stored in a short-term form, like I said, it will be stored in the body for about 3 days worth as glycogen, and that's going to be in the muscle and in the liver. If you have your storages filled, it then gets converted into our favorite thing, fat. And so any extra will go into the production of making fat or lipid droplets to fill up in those fat cells. So what happens to sugar in your blood? Well, it's going to fuel those cellular activities by going through a diffusion and crossing over into those cells. It gets either stored temporarily as glycogen, or if those glycogen stores are full, it will get converted into fat. So those are the 3 fates of sugar in your blood. Carb loading, like we said, carb loading is going to give you energy throughout, and so it's going to actually break down over a long period of time, which will consistently release energy, and so fuel you through a long event. This is a method that lots of runners or people who might be cyclists or swimmers might do. And so athletes can actually double or triple the usual amount of glycogen that's going to be stored in their muscles and liver by increasing the amount of fuel available for extended exertion and delaying the onset of fatigue during an endurance event. When you reduce caloric intake, the body's actually going to go in and it's going to burn this glycogen. So it burns it from the muscles and from the liver. Like we said, you have about 3 days worth. Unfortunately, for every ounce of glycogen, you're going to have 4 ounces of water attached to it. And so for every pound of glycogen you might burn, you're actually going to lose 4 pounds of water. This is where what we refer to as water weight comes in. So at first, when you start a diet, lots of times you'll lose a lot of weight at first. But unfortunately, this is simply water weight. If you eat these carbs again and restock your glycogen stores, you're going to gain all that weight back because all this water has to go into that storage along with the glycogen. And so here's a look at that glycogen molecule. Notice how with each molecule of sugar, you have water molecules attached to it. So you're going to get 4 water molecules. Complex carbohydrates are going to be time-release packets of energy. So it's going to be made up of 1 sugar, monosaccharide units. If you have a disaccharide unit, it's going to be 2 simple sugars joined together. These are things like sucrose and lactose. But if it's a polysaccharide, it's many simple sugars stored together. And so examples of those would be starch, cellulose, and even glycogen. Unlike simple sugars, complex carbohydrates must undergo some preliminary processing before energy can be released from their bonds. So here we have the formation of bonds. We made a polysaccharide or a disaccharide. And once they form into those types of sugars, they're then going to have to be broken apart in order to use them. So if it's a simple sugar, it's broken apart easily and releases the energy. If it's this disaccharide, you break the bond between the 2 sugars, and then you can break up those 2 sugars. If it is a polysaccharide, however, it's going to have to break the bond and then break each sugar as it breaks the bond. And that takes time. So those complex carbohydrates consistently but slowly release energy throughout a period. Starch is going to be around 100 glucose molecules that are joined together. Examples of these are going to be things like barley, wheat, rye, corn, and rice. They're high in starches, even a potato. Glycogen, like we said, is going to be the animal storage form of glycogen, so that's animal starch. Complex carbohydrates, like we said, are time-released fuel pellets. So the glucose molecules become available slowly, one by one, as the bonds between the glucose molecules are broken. And so here's some examples of simple sugars versus complex carbohydrates. With the simple sugars, you see how it's able to break apart and release that energy right away, but then the energy goes down pretty quick. With the complex carbohydrates in this rice, however, the energy production is going to go up once you eat it. However, it will be sustained over time because it's constantly and continually breaking apart these glucose molecules. Not all carbohydrates are going to be digestible, though. You have two types that the human body cannot digest at all. The first type is chitin. Chitin is going to be made up of hundreds of thousands of sugar units linked together in cellulose. These units contain nitrogen and have an increased hydrogen bonding within and between the chains, adding strength. You'll find things like chitin in animals' exoskeletons, like you see here. Cellulose is going to be chains of hundreds of thousands of linked glucose units with hydrogen bonding between parallel chains, and is going to contribute to things like trees' rigidity, and is also going to make cell wall or cells very reliable and very stiff. Fiber is going to be indigestible. It's going to be a plant source that our bodies cannot break down because of cell walls, but it's going to have numerous health benefits. It's often referred to as roughage because it helps swell the fecal matter within the intestines, but as it helps it get larger and swell, it's going to actually kind of clean or scrape the intestine sides. So it helps prevent colon cancer or reduce the occurrence of colon cancer. And so with that cleaning or with that movement, it's going to actually aid the digestive system in keeping you regular and cancer-free. Termites, however, love fiber, and so they're going to play an ecological role in actually breaking down plant material that other things can't break down. Next, we go into our second macromolecule, which is going to be the lipid. Lipids are going to be macromolecules with several functions, including energy storage. So why does salad dressing made with vinegar and oil separate into two layers shortly after you shake it? Well, the reason is because it's going to have two sections to it. One section is going to be hydrophobic, does not like water, so that's going to be your oil. And then the other part is going to be hydrophilic, which is going to be the vinegar. That's water-loving. So with lipids, we have some typical features. They're going to be nonpolar molecules that do not dissolve in water. They're often greasy to the touch and can be a significant source of energy storage. Now, we didn't talk about it, but with the carbohydrate, 1 gram of carbs is going to be 4 calories. So you got 4 calories per gram. With the lipids, however, 1 gram of lipid is going to equal 9 calories. So you're going to more than double the calories from a carbohydrate to a lipid. Now, looking at these, we are showing 3 different forms of lipids. The first one is a fat, and its subunit is going to be a glycerol head with 3 fatty acid tails. So that blue is a glycerol head and the green's the fatty acid tails. Next, you're going to have a sterol. That's going to regulate growth and development. So that's going to make your cholesterols and your different hormones like testosterone, progesterone, and estrogen. And the third type that you see there is a phospholipid. So you'll have the phosphate glycerol head and then the fatty acid lipid tail. And that's going to help function to make things like cell membranes. Fats not only help create things in the body, but they're also very tasty molecules that are going to be plentiful in our diets these days. So like we said, it has a glycerol head region. It's going to have the fatty acid tail regions. And lots of times that subunit for the lipid you need to know is going to be referred to as a triglyceride. And so looking over at the blue at the head and the fatty acid tails. Now the energy is going to be stored between those carbon and hydrogen bonds in those fatty acid tails. Fat molecules contain much more stored energy, like we said, than a carbohydrate. So you're going to have 9 calories per gram with the fat versus the 4 calories per gram with the carbohydrate. Now with these lipids, you're going to have 2 different types that we like to talk about when we talk about food and diet. We have what's referred to as the saturated and unsaturated fats. The difference is going to be the number of bonds in the hydrocarbon chain in a fatty acid. And so if you look here with the saturated fats, they're going to be solid at room temperature, but they're going to have less of these hydrocarbon bonds between them. So the double bonds are what you're looking for. That double bond is what is going to allow it to be liquid at room temperature and to actually be a more healthy fat. That double bond also will give it that little kink in its tail, so it's referred to as a cis tail with a kink in it. So many snack foods are going to contain partially hydrogenated vegetable oils. With this, hydrogenation is going to be something that has become fairly new. This means that the liquid unsaturated fat has an additional hydrogen atom added to it so that it becomes more saturated. And so here you see some girls enjoying their donuts. And of course, these have this hydrogenated oil in them. So hydrogenation is the artificial addition, like we said, of hydrogen atoms to an unsaturated fat. The reason they do this is because, number one, it improves the taste, makes it tastier, and it helps with texture and shelf life, so it lasts longer. So hydrogenation of unsaturated fats is a health problem, though, because it creates what we know as trans fats. These are going to be an unusual orientation of the hydrogen atoms. In normal dietary fats, hydrogen is in an orientation called a cis, so that's that tail that we looked at. Trans fats will cause your body to produce more cholesterol. They raise the risk of heart disease, and then they also reduce your body's production of a type of cholesterol that protects against heart disease, your good cholesterol. So here it shows hydrogenation is going to be that artificial addition, and so you see the difference in the tail. With unsaturated fatty acids, you have that cis that forms, and so it gives that little kink in the tail. Hydrogenation that leads to a straighter hydrocarbon tail shape by converting some of the double bonds into a single bond. And so you look over here, you have partially hydrogenated trans fats. And so you notice these difference in their double bonds. Olestra is going to be a chemical that was created as fairly new. It's a fake fat, and it's going to give food the taste of fats, though, without adding the calories of fat. So it sounds good by helping you take in less fats, so less calories. You're not going to get that 9 calories per gram. But it's also tricking the brain because the brain has been made to actually want fats. So with this one, we're going to have a molecule with 8 fatty acids hooked to a sucrose molecule. So it's hooked to a sugar. It's going to mimic triglycerides and allow fatty acids to stimulate taste buds so that we think we're eating the fat actually. It's then going to pass through the system undigested. Olestra is the only FDA-approved non-caloric fat substitute, and so looking here, it kind of looks like a little octopus versus a regular triglyceride over to the left-hand side. And here's some common foods that you can find Olestra in. So with the summary, characterized by long hydrocarbon tails, fats effectively store energy in the many carbon-hydrogen and carbon-carbon bonds. Their caloric density is going to be responsible for humans preferring fats to other macromolecules in their diet, and also responsible for their association with obesity and illness in the modern world. Cholesterols and other phospholipids are going to be used to build sex hormones and membranes. So not all lipids and fats are going to be fats, nor do they necessarily function in energy storage. The sterols are going to be a second group of lipids that play an important role in regulating growth and development. Looking at these, the top one's going to be cholesterol. Now, cholesterol lots of times gets a bad rap because you hear about it helping to increase your chance of heart attack and heart disease. But it's going to be an important component in cell membranes of animals, and it also can attach to those vessel walls and cause them to thicken if you take in outside sources. This also can lead to high blood pressure, stroke, and heart attack. And so looking at it, you see sterols are all going to be based on the same structure featuring 4 carbon rings. Steroid hormones are going to regulate sexual development, maturation, and sex cell production. They also influence estrogen with memory and mood, and testosterone is going to stimulate muscle growth. Cells in our liver produce almost 90% of the circulating cholesterol. Now remember, I told you that the smooth ER is responsible for creating this within those cells. So that's the organelle responsible. With these steroids, we talked about estrogen. That's a female hormone that helps with memory and mood. And testosterone is often referred to as a male hormone, and it helps with muscle growth. And you have both synthetic variants and natural variants. And sometimes if you take any of these synthetically, they can have extreme side effects. It's lots of times referred to as roid rage. So it will cause you to have a really bad temper. You'll get angry very easily. And also it can actually shrink the male gonads. Estrogen can be found in males. It's simply converted testosterone over into estrogen to help them. And then in females, testosterone can be found, and it's simply made from estrogen being converted over into testosterone. Phospholipids are going to be the major component of cell membranes. So similar to fats, but contain phosphorus and have 2 fatty acid chains rather than 3 for their tails. And then you have waxes, which are going to be strongly hydrophobic, which means they hate water. These are going to be used as a coating on the surfaces of many plants, animals, and insects. Helps keep them from losing water or even becoming waterlogged. So here's looking at a phospholipid. You see it has a phosphate head, which likes water, and the fatty acid tails, which are hydrophobic or hate water. So cholesterol and phospholipids are going to be lipids that are not fats. Both are important components in cell membranes. And cholesterol is also going to serve as a precursor to steroid hormones, which are very important in regulating growth, development. Proteins is going to be our third macromolecule. Proteins are going to be bodybuilding molecules. You're going to see them in things such as wings, blood clots. Over here we have goblet cells creating mucus, heart and muscle cells, and even hemoglobin. So we use them for structure, looking here at the feathers, protection, regulation, contraction, and transportation. The subunit or the small unit of this macromolecule is the amino acid. So it's a makeup of many amino acids that create a protein. There's actually 20 different amino acids. They're going to be strung together with polypeptide bonds, which hook them together, and eventually that makes a protein. They contain carbon, hydrogen, oxygen, and nitrogen. So it contains all 4 of your CHON. Looking at an amino acid, all will have the same similar structure. They all have an amino group, a carboxyl group, and then a side chain. The side chain is what's going to vary. So you have 20 different possibilities of these side chains here, which create your 20 different amino acids. Proteins are going to be essential as a dietary component. It helps with your growth, it helps with repairing things, and replacement. So with growth, you need to be able to build on to what you already have. So you're going to take proteins from your food to actually make muscles bigger, create more hair, repair skin even, repair any muscle damage that you might go through, and to fix things. And you will replace old, worn-out parts with this protein. Proteins also store energy in their bonds. They're only going to have 4 calories per gram, though, just like the carbohydrates. So they're also going to be used as a fuel source. Most individuals need anywhere from 40 to 80 grams of protein a day. However, bodybuilders need much more than that, almost double at 150 grams a day. You're going to have what's referred to as your essential amino acids, and there's going to be 9 of these. Out of the 20, your body's actually able to make 11. 9 of them your body cannot make on its own, so therefore you're going to have to take them in through your foods, and here are the ones that are listed. Complete proteins are going to have all of these 9 essential amino acids that your body needs. Examples of the things that would have all of these are going to be milk, fish, eggs, chicken, and beef. Incomplete proteins are going to be proteins that do not have all 9 essential amino acids. And so that's going to be things such as most vegetables, fruits, and grains. If you consume these, you may be missing out on some amino acids if you don't consume other things with them. So this is why lots of cultures will pair foods together in order to get all of those essential amino acids in one meal. When they do that, they refer to these as complementary proteins. So foods that don't have all the essential acids need to be put together. Examples of these: corn and beans, rice and lentils, rice and black-eyed peas. You have spinach and grains, legumes and nuts. Here's another chart showing you a complete versus incomplete proteins. So apples will have 4 of your essential amino acids. It's referred to as an incomplete. Almonds are going to have 7, so it's still an incomplete. White rice and lentils are getting close, but they're all missing 1, so they're incomplete. And then going from milk down to beef, you see that all of those are complete. So lots of cultural traditional dishes will combine these different proteins together in order to make sure that the person gets all of the essential amino acids. Proteins only work when they are put together properly. So protein functions are going to be influenced by the 3-dimensional shape. This shape is created first by using peptide bonds. So in order to create a protein, your body is going to read a certain section of your DNA, find the gene for the protein that you need, copy it, and then send that gene in an mRNA version to the ribosome. The ribosome is going to read it 3 at a time, which are called codons. From there, it's going to go out in search of an amino acid and bring that amino acid back to the ribosome and start the production of the protein. So this is going to be the primary structure. So it brings an amino acid in, creates a peptide bond, brings another one in, hooks those together with that peptide bond. It continues to hook these amino acids together with peptide bonds until it reaches the end of the mRNA strand. Once it reaches the end, that protein is complete. Then it has to take the protein and make a secondary structure. Hydrogen bonding between the amino acids is going to do this in 2 common patterns. It's going to twist in a corkscrew-like shape, or it's going to zigzag in a folding shape. Then it goes into the 3rd phase, or the tertiary structure. It's going to take those corkscrews and those zigzags, and it's going to fold and bend the secondary structure. Due to the bonds, such as hydrogen bonds and covalent sulfur-sulfur bonds, it's going to help do this. So first you made the chain, then you twisted it or folded it in a zigzag, and now you're going to bend those. Once you've done all that, then you go into the last phase, which is going to be the quaternary structure. So this is when you're going to take 2 or more polypeptide chains that are held together by bonds in the amino acid in different chains and hook them together. An example of that would be like hemoglobin. And so this one over in this picture shows you 3 different polypeptide bonds that they're going to hook together, and they're going to create a functional protein. So at this point, now it's a functional protein. Shape is going to be everything when talking about proteins. So for proteins to function properly, they must retain their 3-dimensional shape, and we just showed you how they got that. When their shape's going to be deformed, they usually lose their ability to do whatever function they were meant to do. So we can see proteins deformed when we fry an egg, for example. It goes from a clear yolk, where you got the yolk inside, and then you've got the clear egg whites. Well, once you fry them up, you're going to denature it. You're going to break those bonds. You reform the shape, and then it turns white. So therefore, that egg white, which was clear to begin with, wouldn't perform the same function. It has been denatured. So that heat is actually breaking those hydrogen bonds and giving the protein its shape. So the protein in the clear egg, like we said, is unfolding. It loses its secondary and tertiary structures, and from there it's disrupted. And that disruption is referred to as denaturation. Or you could say you denatured the protein. So looking up top, we have our normal protein to the left. Goes through some type of extreme environmental change. It could be heat, pH. It could actually even be mechanical. It's going to disrupt the shape and the function of that protein. And so you see over to the side, that amino acid chain has been disrupted, no longer has the right shape. Therefore, it will not perform the same task. You can see this by beating eggs. Their texture goes from being like a slimy egg white structure to actually making like a meringue. That's how we make meringue. We break these. Enzymes can help bring about chemical reactions in a variety of ways. The enzyme lactase, for example, breaks down milk, sugar, lactose into 2 simple sugars that the body can use for energy. So each enzyme is going to have an active site that is going to fit perfectly with a substrate. So that substrate here shown is lactose, and it fits directly into the active site of the enzyme, which will then take and break those 2 sugars apart. So like it acts like a lock and key system. It has to have the exact perfect shape in order for it to work. And then once it breaks them apart, it releases them for the body to use. So we ended up with glucose and galactose, which are both going to be monosaccharides. Activation energy is going to be chemical reactions occurring within organisms that either release energy or consume energy. In either case, the reaction needs a little push in order for it to initiate the reaction, and that push is going to be this activation energy. So enzymes act as a catalyst by lowering this amount of energy that's needed to start the reaction. An enzyme can reduce the activation energy in a variety of ways. It can do this by stressing, bending, and stretching at critical chemical bonds, by directly participating in the reaction, by creating a microhabitat that's conductive to the reaction, or by simply orienting and holding substrate molecules in place so that they can be modified. Sometimes we run across misspelled proteins. So this is when we go in and make an mRNA strand, but it reads it incorrectly, and so it misspells that. When it does that, it creates a made amino acid sequence. That's going to disrupt an activation site and make it so that it cannot break down what it normally broke down. An example of this would be a DNA mistake where it does not make the correct enzyme in order to break down fake sugars. And that would be an example of PKU. So instead of being able to break down that fake sugar, it will build up in the body and can create a toxin in the brain and can lead to death. Here's an example of a mismade or a misspelled lactose enzyme. And so lactase was made improperly. It no longer fits in there correctly, and therefore it cannot break that lactose down into two simple sugars. So in summary, the enzymes are proteins that help initiate and speed up chemical reactions. They aren't permanently altered in the process, but rather can be used again and again. Our last macromolecule is going to be nucleic acids. So nucleic acids are going to be the storage information center. So here we see DNA, and it's going to be made up of three main parts. You've got a phosphate group, you have the sugar backbone, and then you have the nitrogen-containing base, often referred to as a nitrogenous base. Right. So here's your phosphate, here is your sugar. In this case with DNA, it's deoxyribose, and then it could be one of four bases. So here you see adenine, or it could be thymine, guanine, and cytosine. So A's, T's, C's, and G's are going to be your bases. Like we said, nucleic acid are there for information storage. It's made up of individual molecules called nucleotides. That's what we just looked at, which have three particles that make them up. All nucleotides are made up of that phosphate group, sugar, and nitrogen base, or nitrogenous base. There's two types of nucleic acids. You have DNA, which is deoxyribonucleic acid, and you have RNA, which is ribonucleic acid. Both play central roles in directing the production of proteins. The information in a molecule of DNA is going to be determined by its sequence of bases, and those bases are going to be put together, and there's only going to be four of them in DNA: the adenine, guanine, cytosine, and thymine. So together, down at the bottom, you would see an example of a strand of DNA. In order to match it up, you would match it and give it its second strand, which is referred to as its complementary strand. And so you're going to match it with its match. C always goes with G, so you would have C going with the G there. G would be with the C. A always goes with T, and then T always goes with A. So what you would have there would be, and you do it in threes to make it easier. So always try to divide those up into three. So your first would be GCT, and then you would have AAT, and then GGG, and then CTA. DNA is going to hold the genetic information to build an entire organism. So with DNA, it's going to be two strands. We refer to those strands because they're twisted as a twisted ladder or a double helix. So you see the two sugar phosphate backbones spiral around each other, and in the center is where you're going to find your nitrogenous bases that are going to hook together. And they're actually going to use a weak hydrogen bond in the center to hook them together. And so between the A's and T's and A's, it's going to be a double hydrogen bond. And between GC and CG, you'll see it's triple. So we refer to this as base pairing. Always A's and T's and A's, G's and C's and G's. So in summary, DNA is like a ladder in which long vertical element of ladder is going to be made of a sequence of sugar phosphate molecules, and the rungs of that ladder are going to be the nucleotide bases. The sequence of the nucleotide base contains the information about how to produce a particular protein. RNA is going to be the universal translator, so it's going to read the DNA and direct the protein production. It's also going to have a sugar phosphate backbone, but it's actually going to be made of the sugar ribose instead of deoxyribose. Also, instead of having thymine, everywhere it should have put a thymine, so let's say it's making a complement, it read an A, so it should put a T, but not with RNA. RNA is going to put a U instead, so it uses uracil instead of thymine. So the function of RNA is to go in, is to read a section of the DNA, which is going to be the gene, copies that section, makes it into an RNA form, brings it to the ribosome where it's read, and creates a protein. RNA is going to differ from DNA in 3 important ways. The sugar of the molecule, like we said, is ribose instead of deoxyribose. It is single-stranded instead of double-stranded. And of course, we have our uracil, which replaces thymine. It's often referred to as the middleman molecule. So it's going to take those instructions for protein production from DNA and other parts of the cell, and it's going to go to the ribosome where it's going to create those amino acids together and make proteins. So overall, what you should have learned here was what atoms are, their structure, and how they bond. Understand water's features that help support life. We described carbohydrates, their function and structure, lipids with their structure and function, proteins with their structure and function, and nucleic acids with their structure and function. That finishes up for Unit 2, our chemistry unit. I'll see you next time.